Welcome everyone. Welcome to Neuroscience webinar. My name is Rafał Nowak and I will be your host today. And today we'll be talking about transcranial electrical stimulation and functional near-infrared spectroscopy. So I'm very happy to welcome our guest speaker. We have today with us Dr. Mark Muthalib. Hi Mark, how are you doing?
I'm good, thank you.
Good to have you here. And so please let me quickly introduce our speaker. Dr. Muthalib is founder and research development consultant at Silverline Research. He holds a PhD in human neurophysiology from Edith Cowan University, Australia, and has advanced qualification in applied science, technology innovation, management from the University of Queensland, again in Australia. His expertise is in the research and development of neurotechnology solutions to optimize human performance and brain neuroplasticity through non-invasive brain stimulation and multimodal integration with non-invasive neuroimaging techniques that allow to monitor brain state neurovascular dynamics during brain stimulation and cognitive movement and virtual reality performance. So thank you very much, Muthalib, Mark, for joining us today. And the stage is yours.
Well, thank you for the nice welcome, and thank you for allowing me this opportunity to present my research that's been conducted over the last few years, particularly with transcranial electrical stimulation and near-infrared spectroscopy. So as the title would say, is why combine neuroimaging, particularly functional near-infrared spectroscopy with neurostimulation, in this case transcranial electrical stimulation, in research and clinical applications. So I wanted to provide just what is the current standing with publications in the PubMed library, that what are these terms particularly used for transcranial electrical stimulation, which includes tES, which most people would know, transcranial direct current stimulation. There's also transcranial alternating current stimulation, TACS, and transcranial random noise stimulation, and variants that uses a combination of them. But these are the three main waveforms that's used for transcranial electrical stimulation. So the first main one would be tES. The interesting point is that last year there was about 1,022 publications that used tES in either the publication or had original studies. The reintroduction of tES into the research community around 2000 by Michael Nietzsche work kind of increased the understanding of how tES affects neuronal excitability and plasticity. And over the years, the other forms of tES, TACS, and tRNS have also been gaining interest. So for alternating current, there's 142 last year, and for random noise, about 26. So the two main terms would be transcranial direct current and functional near-infrared spectroscopy. So who has actually combined the two technologies together? So there's some initial work in 2009 by Merzagora. There was an offline assessment, and nothing really came about it until around 2013, where I presented some of my work of integrating tES with NIRS. But I initially started this work around 2007. So last year, about 19 papers, and I think there'll be an exponential increase over the next years as the technology becomes more user-friendly and adapted in different labs. So when you talk about fNIRS, what is it actually measuring and where is it measuring from? So if you have a look at the understanding of neuronal activity based on synaptic activity, you have a demand for increased blood flow and oxygen, and you get this increase in blood flow to the specific region that the neural activity is occurring, and this is called neurovascular coupling. So what NIRS is measuring, similar to fMRI, is the hemodynamic response, which is the hemoglobin oxygen levels. So we have a transmitter and receiver. You can have multiple receivers or transmitters. So the light is transmitted at a particular wavelength in the near-infrared light. And this particular wavelength seems to penetrate all biological tissue, and is absorbed by certain chromophores, particularly hemoglobin, oxygenated hemoglobin and deoxygenated hemoglobin at two different wavelengths. I don't want to go into the specific physics of it, but essentially most devices have two wavelengths that are used to differentiate between oxygenated and deoxygenated hemoglobin. So the light penetrates through, is absorbed by the hemoglobin, and the light returns back. And the level of light returning back is proportional to the concentration. And that's essentially what NIRS is measuring, is the light levels through absorption. So more absorption, more concentration of a particular chromophore, hemoglobin in the form of oxyhemoglobin or deoxyhemoglobin. And this is also in the microcirculation, so not in the larger arteries or even arterioles possibly, because it's absorbed at that level. But it seems to be at the microcirculation blood vessels, so around less than 1 millimeter. So this is really at the interface of the neurons and the vascular system. And this will be considered part of the neurovascular unit. I'll explain this in more detail, how tES is really affecting this whole system together. So what do we actually see in a NIRS response? So in the bottom left, you see this increase in oxyhemoglobin and a smaller decrease in deoxy. Now, why does this happen? So most people would imagine you have increased cerebral metabolism for energy production, and this is the neurometabolic coupling. And in this case, you have a decreased oxyhemoglobin because the oxygen is used, while you have an increase in deoxy because there's a larger concentration of deoxy, because it's now after the oxygen has been consumed or taken out from the hemoglobin, there's a higher concentration. So this is what you'd expect if the NIRS is purely picking up oxidative metabolism. But it also is measuring the neurovascular coupling, which is cerebral blood volume and cerebral blood flow. Now, they both have slightly different ways of representing oxy and deoxyhemoglobin, but essentially with cerebral blood flow increases, you have a large increase in oxyhemoglobin and a large decrease in deoxy. And this is considered like a balloon model. So with a large increase in oxyhemoglobin compared to a smaller increase in deoxy, the whole volume shows a decrease in deoxy. And that's why you see this decrease in deoxy occurring. So overall, we see this increase in oxyhemoglobin and a smaller decrease in deoxy. And this is traditionally what functional MRI bowl signal represents. So one of my forays into understanding how NIRS is represented with other technologies, we worked with Dr. Muthalib, who was originally in Kiel. So we were able to combine fMRI with fNIRS and EEG all at the same time. So we did a finger tapping task, and we had the subjects do 10 blocks. So each of these, on the right panel, you can see each of these dashed lines represents a 30-second block of tapping with a 30-second rest. Now, if you look at the gold standard of the fMRI, you see this increase in the bowl signal and a decrease after you stop. And this happens over the 10 blocks. So with the fNIRS, so we can see on the image on the left side, overlaying the MRI scan, you can see where the NIRS probes were on the scalp, and the theoretical time of flight, or the theoretical flight of the photons over the tissue and where it's measuring from. And essentially what we see is this oxyhemoglobin increase with the task period and a decrease in the rest period over the 10 blocks, which is quite similar to the fMRI. And with the deoxy signal, we see this decrease in deoxy occurring with the task period and increase occurring in the relaxation phase. So this is quite consistent with fMRI as a gold standard for functional activation measurements from local regions. Also with the EEG is slightly different. We use EEG and EMG coherence, the dynamical coherence of the two techniques, to represent that in the task phase, you have this larger coherence occurring between the two. And this is also quite consistent over the 10 blocks. So this is kind of some initial pilot work I presented in 2013. And the full paper, which was from a PhD student working on the project, Anwar, and the main title, which most people would see, is the effective connectivity modeling of the data. But in the supplementary materials, you'll see this data, which was not necessarily so new, but I like to present the work because it shows the what the group level changes were. So over the time periods, we have the fMRI, the top row, and each of the different colors represents the region of the brain that the NIRS is measuring, or the fMRI is measuring from. So what we see with the fMRI is this increase in bold over the 30-second task period. And for 3 different movement tasks, so simple finger tapping, simple finger sequence, and complex finger sequence. I still know how to do it. So what we see is this increase in oxy and decrease in deoxy. And this is a group level changes. So quite nicely represented between the 3 techniques. The EEG was also split into scalp and source level. And these are the correlations. I won't go into specific details, but essentially they were quite well related to each other. So for me, it confirmed the use of NIRS to at least measure from the cortical layer on the brain for different tasks, particularly in this case, the movement tasks. So a major point of that work was the neurologists I was working with were interested to use NIRS for deep brain stimulation patients. So they have electrodes deep in the brain. fMRI is not really recommended because of heating of the electrodes. So they wanted to see if NIRS could be used instead. And with that study we did, they were quite happy to allow us to do some pilot work on some Parkinson's patients with deep brain stimulation. So essentially we did a cognitive task, go-no-go task, and had the patients with stimulation on or off and do the tasks. So just, I want to be quite brief. We found a reduced activation with the stimulation on in this case. So here we can see how the NIRS needs to be incorporated into different EEG setups, and that's something I will discuss a bit later. So the other work that got me into TDCS or TES work was muscle stimulation, so neuromuscular electrical stimulation. So here we do very similar work where we stimulate the peripheral muscle and nerves. And this work originated similar to TDCS from the fathers of neurophysiology, Galvani, and further work from Duchenne, using different batteries to stimulate different nerves in muscle of the head, for example, for facial expression. So this neuromuscular electrical stimulation, similar to, we have 2 pads and we stimulate different muscles. And the idea for me was, I was working on measuring muscle activation. At the same time, I was interested to see how does it affect the brain, because what we have is this stimulation of the nerve, which is mixed. So you have both sensory and motor axons. So sensory axons then traverse back through the spinal cord and back up to the brain, where it's sensed. And most people would know the sensations they get with any electrical stimulation. It's sensed, but this also then modifies how the brain actually integrates sensory and motor integration. So this is some of the work I was doing back in Australia during my PhD work. So we had the muscle electrical stimulation, muscle NIRS, but we had a probe on the forehead. So Professor Kazori Nozaka was my PhD supervisor. So he was a good model here for me. And also when we got the results, I was working with Professor Marco Ferrari, who's one of the godfathers of NIRS in general. And we were wanting to know what is this large increase in oxyhemoglobin that we saw during the muscle stimulation. So we also looked at increasing the current intensity. And this is what the normal procedure for muscle stimulation, to get as much muscle contraction. Without confusing people too much, when you do muscle NIRS, you get an opposite response for oxy and deoxy, because the blood flow is limited with the contraction. So what we have is oxyhemoglobin decreasing while deoxy increasing, compared to the brain, where it's the opposite, where there's a larger blood flow to demand. So what we have is that from the initiation of a certain percentage of the muscle contraction, we're stimulating at near maximum thresholds. So we can see that, I mean, compared to tES, we're going at, you know, around 30 to 90 milliamps. So very, very high levels. Pain sensations are there, but we can keep increasing the current intensity. There's been no skin damage I've ever noticed. It's just the sensations of pain is quite high, but it's also modulated over time. So you can keep increasing the current. And what we noticed was in the prefrontal area, this large increase in oxyhemoglobin. So one of the things with Professor Ferrari is, what is this large increase representing? And this is also something that is relevant for tES work that I'll present a bit later. So we wanted to further investigate how NIRS could be used in a multi-channel setting. So working with Professor Perret in Montpellier. So we embarked on this large project with an Italian group with Professor Ferrari and Torricelli in Milan. So we looked at comparing the NIRS system that Stefan had, which is a continuous wave system with a time result system. I'll explain that in a second. So we set up a similar experiment where we do wrist extension. So you can see the subject here, and we do voluntary contractions and electrical stimulation contractions. And we look at the brain response. So we see this nice contralateral hemisphere activation on the left panel, a smaller increase on the ipsilateral and the prefrontal as we increase the currents. This is how you would represent in a group analysis over time, so over the 30-second blocks. So it's a 20-second blocks of stimulation with 5 seconds rest. You see this large increase occurring during stimulation, and when you compare voluntary to stimulation. So this was the second part of that project, where we looked at separating the skin from the cortical layer. So using near-infrared, we're using time-resolved NIRS, where you can look at the photon time of flight. And the model that Professor Torricelli had developed was, if the photons return back quickly, they have traversed a shorter distance on the skin, while if they come back later, that would mean they've gone further deeper in the tissue. So what we were able to see was, if you take out the skin or the superficial, we get this nice activation as well. So overall, we see the cortical layer was there underneath the superficial layer, and even sometimes a larger activation in the cortical layer was able to be differentiated. So that's leading on to the work I started in Montpellier, where we were able to then continue on stimulating the brain using transcranial direct current. So just some history of the technique using batteries. So goes back to Aldini in 1801, treated a patient for depression, essentially, using this setup on the right. At least he documented this. Other people have been using it throughout the millennia. Probably not well documented, but this was a well-documented case for curing depression using a simple battery. And Michael Nietzsche in around 2000 did further investigations using anode cathode tES direct current, and measured the brain excitability using TMS as a standard measuring method, and showed increases in excitability. So this revamped the use of TES in research. So the bottom one would be my montage that I set up in Montpellier, combining NIRS with tDCS and EEG. Again, this was a really research setup using the available technologies we had at that time compared to what Neuroelectrics and tES provide a nice wireless system that you can use relatively easily than the way what we had to do here. So just some background to mechanisms. The traditional way has been that with an anode cathode pair, anything underneath the anode will be depolarized or higher excitability, while under the cathode would be hyperpolarized or reduced excitability. But with the configurations of neurons, they lie in different orientations, and the orientations affect how the polarization of neurons at different parts of the neuron itself. Of course, when you look at a larger scale of connected neural circuits, it becomes very complicated when you consider anode and cathode and try to differentiate the two. I would have a look at this nice paper in Nature Communications that describe the complex nature of the excitability of neuronal structures as the traditional way to understand how tDCS affects brain excitability. So when you talk about optimizing tES protocols, in traditional way is open loop in the sense where everybody gets the same dose. So you can look at the current intensity. We can go at 0.5 milliamp to 4 milliamps now, as in clinical settings. So, but everybody gets this same amount. Of course, we can change the waveform from, this is a traditional direct current stimulation where you have the random noise and alternating current on the bottom right. Also, you have a pulsed waveform too. And the duration of stimulation, some of the work of Michael Nietzsche showed around about 10 minutes seems to be optimal, about 13 minutes. And of course, people have considered a little bit higher, 20 minutes would be better, but that's not always the case. There's always this effects on excitability, and duration not necessarily would have a better effect. Of course, the electrodes and montage fit hand in hand of what you can, where you can place the electrodes to affect a certain area of the brain. So when I went into using tDCS, we were doing neuromuscular fatigue experiments, and this is around 2007. So when I heard about the use of tDCS for fatigue research by Koji Manian and Priori in Italy, that came around the same time when I was looking into this, I wanted to replicate it, and this is the montage they used: an anode on the motor cortex and a cathode on the shoulder to reduce any confounding effects of the location of electrodes. So we were not able to show any modulation of neuromuscular fatigue as assessed using an isometric task for a certain period until they couldn't hold it. So if you do one session, you wait 1 hour and you do tDCS before the second session, we didn't see any change in the time to fatigue. But the difference is that we used young, healthy men, so their residual fatigue might have recovered by that time. But the other aspect was I was trying to put where I can put the NIRS. So ideally would have been around the motor cortex. But since the electrode size is larger than the transmitter receiver distance, which needs to be around 3 centimeters, I wasn't able to find a solution around it. So also we were looking at the prefrontal area. So we put an area that's away from where the stimulation site is. And interesting thing was that we were able to show that as the person was getting fatigued, and at the point of fatigue, this point, the NIRS response, or the amplitude of the NIRS response was very consistent at the point of fatigue. So other work of Stefan in my lab here have used this metric in other exercise protocols. So I embarked on how to integrate tDCS with NIRS around 2012, where we were able to get working with Neuroelectrics. One of their first devices came out at that time. So I tried to integrate here the first picture with the tES device with a one-channel system on the forehead. So they seemed to work well. They weren't interfering with each other. The EEG, of course, gets affected by the electrical stimulation, but the NIRS is not affected by it. So we're able to get the same NIRS signals whether the stimulation was on or not. So that confirmed to me. I then formed a collaboration with Professor John Rothwell, again, godfathers of electrophysiology. And he was interested to guide me on solutions. And at that time, the high definition montage came out around 2012, 13. And we were looking at how to get this integrated into a single cap where you can put the NIRS around the smaller electrodes. So the solutions we also had to find was how we can place the electrodes and where we can place the NIRS, understanding the geometry and the constraints of this transmitter receiver distance. So this was the setup we had in 2013 with essentially a cap from hairdressing, but then we had to modify it further, just using the SNARSTIM EEG system, and then we integrated further with another EEG system to get more channels because we only had the SNARSTIM 8 at that time. But of course, Neuroelectrics then have the SNARSTIM 32 that we don't now need another EEG system. So just some background to what is high definition tDCS. Modeling studies essentially have shown that it focuses the electric field close to where the anode is located, compared to the traditional large pads, which has a diffuse electric field. So this is modeling. This is using finite element modeling of different structures and to see where the current would flow based on the resistance of different tissues. A validation of this model, of this method with Dylan Edwards using TMS, as well as a high voltage tES montage, which is quite painful, but essentially he was able to show that if you move the whole setup from the front to the back, where the highest level of MEP, the excitability levels or the motor evoked potentials, were in the region where the anode was in the center of the motor area, C3 area. So this shows quite nicely that the electric field is focused in this region. So that gave me a platform to develop my protocol, my setup, my montage. So this is kind of a first pilot work or preliminary work we did. We have two phases, two interesting discoveries. One thing was we did a finger tapping task. And on the left panel, we can see if you do right hand movements, you can see this left hemisphere. If you do right hand task, left, you can see this large increase in channel 4. I've highlighted that. And the opposite hemisphere, we have a smaller increase. So there's bilateral activation. The two hemispheres are working together to do this simple finger sequence task. So this is before and during stimulation. We found that both hemispheres reduced activation. And that's something that we wanted to explore further. Another discovery we found was that during the stimulation, you can see that channel 3, 4, 5, 6 on the bottom right panel, we see this large increase in oxyhemoglobin and a smaller decrease in deoxy occurring in these channels where the stimulation electrodes were. Now, these responses are very similar to those responses I showed you with the peripheral stimulation in the order of about 6 micromoles, compared to the normal activations about 1 micromole for task-related activation. So we also looked into this further. So I'll describe first the movement-related fNIRS activation and modulation with tES. So the full study was done using the SNASTIM 8. So we had 4 channels. So the blue would be the cathode and the anode in the center, C3, and the NIRS 8 channels on the left side and 8 channels on the right hemisphere. So essentially what we see is this increase when you do a right-hand task, with this increase in oxyhemoglobin, decrease in deoxy in the left hemisphere channels, and a smaller increase in the right hemisphere. And what we found was that when you did tES online while you're doing the tasks, we found a reduced activation. So we considered that this could represent some form of a greater efficiency. So you have to do less work to get the same movement task. Now, an issue with this particular setup was that we didn't have the offline condition by itself without doing an online condition first. So we work of a PhD student, Pierre, continued the full experimental protocol where we had 3 groups: an online group where they did the task during the stimulation, an offline group where they did the task after 20 minutes of stimulation, and a sham condition where there's a half and half of both. So here on the left side, you can see the setup. In this case, the right-hand task, so finger sequence, while doing the stimulation. So similar to a preliminary study, where in this case, we're using the deoxy signal. So what here, an increase in deoxy would mean less activation. So it's counterintuitive to what we see with the oxyhemoglobin decrease. So essentially what we find is during an online stimulation, when you do the task, we have a reduced activation. So that's confirming what we found in the preliminary study. But about 30 minutes after, we found a rebound in activation. So we considered this to be some indication of plasticity occurring. Again, we didn't do any further measurements after this. So we also wanted to look at if you stimulate, as we said in the previous, that we found bilateral activation effects. How does stimulating one hemisphere affect the opposite hemisphere? So working with Dr. Wei Peng Teo, who's now in Singapore. So we had looked at some cross-facilitation effects. So if you do an isometric contraction of one arm, and if you measure the opposite hemisphere, you see this increase in excitability of the opposite hemisphere. So we wanted to see if this can be modulated. So we did stimulation on the right side, and we did a task using the right side. And what we found was the left hemisphere increased excitability with this high definition montage. So even though the electric field is focalized in the area of stimulation, it does have its connections with the opposite hemisphere in this case to increase its excitability. So some other aspects of what we did in this particular overall project with Pierre Besson's PhD work was to see a circular drawing task. We manipulated excitatory and inhibitory stimulation, and but essentially what I wanted to show here was that when you look at a circular steering task where it's a speed accuracy trade-off, that tES or anodal stimulation seems to increase more the speed of the task. Now, this is important because this is something that we're looking at for stroke patients, is something that can enhance their speed of movement, which is a motor cortex role in speed of movement. So essentially, we're able to show some preliminary evidence in healthy subjects that we can increase the speed of the task with stimulation over days. So it's not just one session, but this is over 3 days of training. So it's quite a large increase we occurred. But of course, errors were at the same level as sham. So going back to this other interesting observation, the bottom right, which was this during stimulation. So this is kind of my interest to see what is this representing. So with this collaboration we had with the Polytechnic of Milan, with Professor Torricelli, we were able to do comparison of the studies we did in Montpellier with the Milan. One subject did the same experiment, and we can see that when you look at the NIRS response from the normal NIRS system compared to the time response system, you can see that is a similar levels. So when you did this correction for the superficial layer correction, we see a more traditional increase in oxyhemoglobin at the levels of normal activation. So underneath this superficial activation and the causes of this, we see that the brain cortical level activation is also increased. So, so underneath this large increase is really cortical activation that NIRS is measuring during stimulation. So another interesting aspect, as I said, we showed that the channels in the center were larger than the channels in the outside. So so we did some further analysis of the data, and we found that when you look at the oxyhemoglobin increase in the channels that are inside this high definition montage compared to the ones outside in blue, you see this large increase compared to the outer channels. Again, there is some effects of either because the receivers of the NIRS system is in the border, so there's contamination from each other. So it's not, you can't say one is really outer and one is really inner. There's a mix, but essentially we can see the difference between the 4 channels on the inside to the 4 channels on the outside. And the metric we used was the area under the curve over the full 600 seconds or 10 minutes of stimulation. And here on the bottom panel, bottom row, you can see the differences. We did 2 sessions of tES, so they were quite consistent. And both the inner and the outer channels for the both sessions were very consistent. So we wanted to further explore this because the contralateral hemisphere that wasn't stimulated had no observable changes occurring. So we can then validate that the NIRS was able to pick up this electric field, focal electric field, that was shown by Dylan Edwards we're using TMS and high power tES. So we looked at see if machine learning can also classify if a channel was if the fNIRS signals were coming from a channel that were in the inner or outer channel. So with Dr. Nicola Sutton-Shirani from the lab here, we did some preliminary work. Again, this is quite preliminary, and essentially what I wanted to show was that we were able to show machine learning could also classify this inner and outer channel from just the NIRS signal of a 30-second block. And with these accuracies levels, with different models, machine learning models, we can be quite confident that the NIRS signals themselves are providing information that can be used eventually for optimization methods for tES. We're looking into how we can use this type of machine learning methods for this purpose. In parallel, working with Dr. Anirban Datta, who's now in the UK, in Lincoln, some of the initial work, he was collaborating with us here in Montpellier. We're looking at modeling work and looking at the neurovascular units. So one of the first investigations we did with a student of his was to look at the relationship between the NIRS and EEG, in this case, using an autoregressive model to look at the interactions between neuronal and hemodynamic signals. So in this case, how does stimulation affect the 2 signals, EEG and NIRS? And are there relationships between the 2? Further on from this, trying to explain how the interactions between the interaction between the hemodynamic and the electrical signals, is to understand that when the vascular system itself is innervated by peripheral nerves, which has vasodilatory capacity. So in this case, the outer PL arteries have innervations of the blood vessels that, similar to the peripheral nerve stimulation, if they're modulated, would also increase their excitability and release vasoactive substances that can then increase blood flow, and then that perfuses through to the brain area, and that would also then cause an increase in neuronal activation. So in this case, we have the different structures of the peripheral nervous system that supply the outer layer, while the intrinsic innervation. So again, these nerves are surrounding the blood vessels and surrounding the neurons themselves that are important for the modulation of tES, but you can't overlook their effects themselves. So the work, again, with Anirban's student and Dr. Shabjit Roy in India, they went to some painstaking modeling of different neurovascular unit structures to look at this increase in total hemoglobin, which is oxyhemoglobin and deoxyhemoglobin combined, as an indicator of blood vessel response. So looking at different modeling, so use the gray box modeling method, we're able to determine which structures contributed to this real physiological response that we're measuring. And from the models that they developed, they considered that the perivascular potassium seemed to be the best model to predict this increase in total hemoglobin. So again, what is the importance of different structures in the neurovascular unit being modulated has also recently gained more attention, with Meron Bixham and his group coming up with this nice review paper that puts it into perspective, all the structures that could be modulated in the region where the stimulation has its electric field. So it details quite clearly mechanisms and modulation capacity. So I would recommend to read these papers to understand this concept of neurovascular modulation. So before, when we used NIRS, it was always a neurovascular coupling. So neurons increasing the excitability, that increased demand, and the blood flow increase. But in this case, we have the neurovascular unit structures, including the peripheral nerves surrounding blood vessels, being modulated, that increases blood flow, that then increases neuronal activity. So it's a mix of both. And this would be the representation of what NIRS can provide, is a direct assessment now of neurovascular modulation rather than a secondary aspect. So this is something that gives me credence to further do research on this, that we can actually get a direct measurement of TDCS or TES effects on the brain. How are we doing for time?
You still have some few minutes, Mark.
Okay, so just wanted to give just some other work that we've been doing with the StarStim NIRS integrated systems. So this is kind of some work we did with Roy Cohen-Kadosh in University of Oxford, where we integrated a StarStim 32 with a Bright 24 eventually, and we looked at how we can measure and modulate arithmetic learning. And essentially the work showed that you can enhance your learning or plateau learning with this type of transcranial random noise stimulation during the learning process. But the other aspect is that this is the NIRS measured during learning phase, for example. So each of these lines would represent when an event would occur, to when a mathematical combination was supposed to be solved by a subject. So as you can see, when the event was presented, the thought patterns is in the prefrontal channel was increased, and then once they gave the answer, it decreased slightly until the next combination was to be solved. So within, this is an event-related NERS response, but we initially were looking at a block design. So over the full 8 blocks or 8 different calculations. So this is one way that, you know, with the EEG was contaminated, but there's a ways to overcome this. But at least with the NERS, we can get the values at the time of measurement without much pre-processing steps being used. So this is some work I'm doing with Professor Javier Analasta in the University of Deusto in Spain. So looking at enhancing creativity. So we're using, in this case, tES on F3, F4, anode on the right side, cathode on the left, and we had the NERS. This is a Octomon Plus with a StarStim8, and essentially this is 4 channels on the left and 4 channels on the right side. So this is during a stimulation session. So you can see that without any filtering, you can see the heartbeats within the signal, which the RTN system can clearly measure. Each undulation of the heartbeat shows a very good probe to scalp contact. And what we see is that over the different conditions, we see an increase in activation and increase in activation by oxy increase and a deoxy decrease occurring. So again, we're looking into the results from this particular study. Other aspects is the combination of VR with tES. So this is a big project we're doing in Montpellier with the Centre Hospitalier University with Dr. Karima Bhakti. Here we're doing both NERS and EEG are used before doing a VR and stimulation protocol over 3 weeks, and then post-assessment, and then 3 months later. It's quite a comprehensive project, clinical trial, still going on. It's about, we're past halfway. Essentially the montage we used was a StarStim8 with a Octomon Plus to look at, you know, more proximal muscle regions. So you can see the NERS is located close to the midline with 8 channels of EEG to do the reaching task and a circular steering task, similar to the one we presented earlier. And all this is all synchronized data with the kinematics, with the NERS, EEG, all synchronized, you know, using LSL. So it's quite nice. We can get all the time series data aligned and analyzed in one data set. The other aspect is that we're able to do stimulation and measure the NERS and EEG during the VR sessions. This is quite exploratory, and we're looking into this data once unblinding is done. So we'll be able to see which subjects were able to recover better, and if that is represented by their brain activation during the games. It's a form of precision medicine that hasn't been done before with regards to stimulation and using NERS and EEG in this context. So some preliminary works, not preliminary, but parallel works by the same group with the PhD student Camille Muller, where we're looking at the reaching task and the circular steering task. Essentially, we're able to show a difference between healthy young and healthy aged. I don't want to go into specifics. I'd recommend reading this publication. But we're able to get both NERS and EEG synchronously measured and analyzed to provide a complete picture of what's happening in the brain when you do these particular tasks of reaching or circular steering, which is a performance assessment. So the future of optimization in a case where, how can you use modeling methods, which is becoming the standard, is where people are using different MRI scans or MRI scans of patients, of subjects, doing finite element modeling to determine ideally where to stimulate and where to measure from. And what we're looking at is having this optimized pre-stimulation method can be further enhanced, further optimized, having an online assessment. So in this case, whether we can use NIRS to then validate if a pre-stimulation optimized protocol really is optimized while they're getting stimulated, and then slightly modify the parameters in a closed loop fashion. And this is the kind of context we're looking at. Is we have an optimized protocol with modeling methods. Neuroelectrics provide this service where you get an MRI scan, they provide locations of stimulation, the currents, and then where we can apply the imaging during the stimulation to then check to see if this patient or this subject might need to increase the current intensity or modify the duration based on the data that we're seeing. So this is what I consider would be the future of the technique. So these are some of the integration solutions we've set up at Silverline Research that will support research labs to look at different areas of the brain, to measure from them and then stimulate, so giving guidance on this. And as well as we have some we have software that you can use for training. Again, this is more high level closed loop, but it's nice for people to see how you can start to do closed loop stimulation, as well as getting some online analysis of the EEG and the NIRS signals in real time. So this is a service provided if people require from Neuroelectrics or tES. So I wanted to do a demonstration of the OptiStim software running simultaneously with the Neuroelectrics and the tES OxySoft. So both the devices, both the software is running, and the OptiStim is sitting, essentially viewing the data from both systems, and then using different methods to do online analysis, as well as providing some closed loop stimulation protocols. So I wanted to show it this way because I can probably talk you through what's happening here. So this is the preferences screen in OptiStim, where we can link with the Neuroelectrics protocols directly. So you can choose the protocol that you want. Tells you the channels of EEG and channels for stimulation based on the Neuroelectrics. And if you want to, when you integrate it with the NIRS, it can look at what the frequency of the NIRS is being sampled at. In this case, you have to go into OxySoft and choose the LSL stream. And that's what's happened here. So this is a real life demo of when I was measuring myself with a StarStim 8 and an Octomon Plus. So here we have both systems running. So this is some of the back end of the OptiStim, where it's telling you what it's doing. So from here, you can do a display of both the Neuroelectrics EEG, as well as the tES OxySoft signals, where it's going to stream the data. And on the top right-hand panels, we can get some online metrics. For in this case, from the NIRS signal, we can get heart rate. And from the EEG, we can get a center of gravity assessment of the EEG channels. So once the system is communicating with the Neuroelectrics NIC, it then allows for the two systems to pair up for safety reasons. And then the data is transferred through into OptiStim. And here you can see on the right-hand side, heart rate monitoring, as well as the EEG metrics. I have to say that I didn't place the NIRS and EEG optimally because I just put it on my head at that time. So this is kind of very bare NIRS and EEG preparation, but most of the channels were providing some good heartbeat signals for the NIRS signal. So this is the streaming feature. So in the next option, we have where you can select different designs. So the design screen, I think here we're able to show that you can get the level of oxyhemoglobin on the right-hand side, the top right-hand panel. So you can change this. You can get different power, relative alpha, theta, and this is measured online as you are doing a measurement. So you can get an understanding of what the subject's resting state metrics of EEG and NIRS are. So the other aspect is doing a stimulation and NIRS and EEG simultaneously. And in this case, we can change the design. And here, what I've set up is an entrainment program, which essentially is measuring the EEG for 10 seconds and then stimulating. Sorry, 20 seconds of measurement and then 20 seconds of stimulation. So again, it's not a clear sinusoidal stimulation for the alpha frequency, because it is measuring directly from my head. So so you can see that it's stimulating, measuring, stimulating, measuring. So you can see how the stimulation is affecting my alpha level, alpha band, and then stimulating at that frequency, well, how that affects the brain accordingly. So it's in a closed loop program. Another program that we've set up is a conditional program where you can use NIRS in this case. This is mainly using EEG, the entrainment. So where we can look at the EEG and NIRS responses, and at the end, there's a result screen that can provide the analyzed data post-processing, but it's done immediately after the session. So you'll be able to check this as well as get the raw data and do your own analysis. So you can quickly see how well a protocol is running with this software. So this is the conditional program where you can select some rules. So in this case, we can choose a NIRS channel, say oxyhemoglobin channel 3. If it increases by 3 micromoles for more than 3 seconds, then the stimulation parameters will be modified. So then we go into the display section, and you can I think we'll go to the, so here the display, and then you run the protocol. Okay, so here we're running the protocol. So this is the conditional protocol, which is looking for a NIRS increase of 3 micromole change. So I had to then focus on calculating in my head to increase my NIRS response, oxyhemoglobin levels, above 3 micromoles. So this is you can look at the middle panel, the top middle panel, when after the initialization protocol phase. The middle panel will show the level of threshold that I have to get. So in this case, I still haven't reached the threshold. So I'm doing a calculation to increase the level of my NIRS response. So the stimulation won't come on until the threshold has been reached. So I'm calculating. So here I'm calculating two large numbers. And once the stimulation is reached, there we go. Then the stimulation will come at 1000 milliamps, microamps at 20 hertz frequency. So then the NIRS goes back down, and then the same process occurs until the threshold is reached again. Okay, I think that's it. If people have any questions?
Yes, thank you very much, Mark, for this excellent presentation and demo. So we are ready actually to take some questions. We have a question from Lin Cho. Thank you for the interesting talk. And the question is how you deal with the possible bridging issue between tES and fNIRS electrodes, given the existing neurovascular modulation mechanism. And I think this question regarding simultaneous recording of tES and fNIRS.
Bridging in the sense of interference? Is that
of interference and the electrodes, touching of the electrodes, and I guess being close, the stimulation electrodes to the transmitter and receiver. Yeah.
Well, the tES probes that I've tested on haven't caused any interference. Again, the probes themselves are still a distance apart, but there's no discernible electric field produced by the transmitters of the NIRS to cause any interference. There's really no bridging I've noticed.
Okay, and so in any case, with fNIRS, you are dealing with some signal to noise ratio parameters and values, and how you deal actually in terms of interpreting the fNIRS data, especially if you're going to the individual level of subjects, of patients.
Yeah, I think I didn't want to go into details about the pre-processing steps, but of course, once we have this nice heartbeat signal, and we know that the probe is in nice contact with the scalp, then we're sure that the signals should be not interfered by the noise of the hair that is usually a problem of having to move the hair. But if you get these nice signals, then other aspects are movement artifacts. There are methods that we use for detecting the movement artifacts. So a common tool people are using for analysis of NIRS is the HOMA toolbox. And there's many different scripts that look into detecting when an artifact occurs based on different thresholds of changes, and then interpolating that section out if the baseline is shifted, things like this. I mean, ideally, when you set up an experiment, once you have the cap nicely set up on the head, it's secure, there's not much movements occurring. We try to, of course, there's movement possible, but we want to keep minimizing as much large movements. So, of course, NIRS is much better than fMRI. fMRI, you can't do any kind of movement at all. But here you can move, but the whole system needs to be well kept together to minimize these movement artifacts. But there are methods in this HOMA toolbox. If you look at the tES website, they go through the different toolboxes that people can use to go through the signal to noise problems and go through the analysis pipelines. And this is some other aspects of the research service that Silverline provides, is to go through this state. Once you set up the cap, set up the the analysis and then work through this process, particularly for labs that haven't got the strong NIRS background.
Okay. Thank you. So actually, not so. As we know, all techniques, even like fMRI and EEG, they have some advantages and limitations. So this is also related to fNIRS. Actually, there is a question from Eugen regarding OptiStim software.
Yes.
That you were showing. So it's Eugen is asking if there is a possibility to get the link to the software, or it's a proprietary software, and how someone could get access to the functionalities you were showing.
So at the moment, currently, it's proprietary software that we developed to just showcase the system and to show the possibilities of the integrated system. Eventually, we might be able to provide this as an independent software, but currently it's linked with the Neuroelectrics StarStim setup, and for also reasons of safety, because the stimulation protocols within the NIC have to be maintained quite well. So this is currently only provided through the support services when you have a system, or if somebody wants the support service that includes the training in OptiStim, that can be requested through Neuroelectrics or even tES to provide a training on this, where you'll be able to test it with your system if you have already the system.
Okay, so actually the best way would be to contact you at silverlineresearch.com, and then you will provide more information.
Yes.
Okay, perfect. Another question is regarding the montage. Actually, some of the montages you were showing of fNIRS. If do you think that also other brain regions, in example, parietal regions, are appropriate for hemodynamic registration, or the montage is basically frontal?
Well, the different NIRS cap layouts, you can have a whole head. It's not limited by what I showed. The systems that, particularly the wireless systems that our tES have, I've tested those. They've got a 27 channel montage, but you can put two of those systems together to get double the amount. You can get a full head coverage. So of course, yeah, we can, but then that also limits the EEG capacity too. So it's how best to solve this problem of locations on the cap. There's only so much space that you can put a probe on, considering that each probe takes up a centimeter or two of space on the cap, or circular space. So we need to configure the locations. If you use the EEG locations, then we are only able to use the locations outside those locations.
Okay. Thank you very much. Another question.
Yeah, of course, it's possible to measure from any area of the brain.
From other regions, yeah. Technical question regarding virtual reality. If the light from VR screen leads to interference in fNIRS mechanism and registration.
So I think, would that be a more headset?
Yeah.
I haven't tested it fully, but I know that our tES have used their NIRS system in VR systems proper, and I haven't been informed that there's been any interference. Again, this is something that I would look into in further integrations in the future.
Okay, and there is a question regarding comparison of artifacts coming from the parietal regions and frontal regions. If there are any differences between artifacts that you can expect from the parietal regions or frontal regions? In terms of
the artifacts are purely, if you do move, purely if a probe moves on the scalp, I don't expect to be different. It's purely a case of head movement affecting the probe on the head. Unless, it's purely that those are the artifacts that you're normally seeing. It should be a general artifact for both frontal and the parietal, like if you're doing walking, for example.
Okay, okay, perfect. So this was our last question. Thanks, Mark, for your presentation. Thank you, everyone, for all your questions, and obviously for joining us today. So I hope you will join us again for the next webinar, and this will be on 31st of August with Dr. Daniel San Juan. And Daniel will be talking about transcranial electrical stimulation in epilepsy, so applications of transcranial electrical stimulation in epilepsy. So please visit our webpage. You will find there the registration page, information about any future webinars that are coming. So thank you very much. Have a great day and see you next time. Thank you, Mark. Thank you. Thanks a lot. It was amazing. We've learned a lot about NIRS, about EEG, about
I hope so. I hope.
simultaneous
People, please email me
stimulation.
if there's any questions. Yeah.
Yeah. So if there are any additional questions, you can always reach to Mark, and Mark will be happy to answer any additional questions.
Okay.
Thank you.
Bye-bye.
Thank you. Bye.